that brings us to capillaries. We said that capillaries were the smallest vessels that we have. Again, remember, some capillaries are smaller than one red blood cell. Okay, so they're very, very little vessels, and they have very thin walls. When we look at capillaries, again, remember we said that the walls of capillaries don't have the tunica externa, they don't have the tunica media, right? It's just that little tunica intima. They're very, very thin walls. Capillaries permeate all active tissues, kind of. They're at least near all active tissues. Remember that epithelial tissue is avascular, right? So epithelial tissue does not actually have a capillary network in it, but it'll have it right near it, right? Like up at the very top of the dermis, remember we saw capillaries? Right near those, the epithelial layer in the epidermis. Also, we'll see that in some cartilage. So when we look at cartilage, we'll see that inside the cartilage in the center, it's avascular, but the surface is covered with the perichondrium, if you remember from tissues, that really, really vascular membrane on the outside. So those are my two kind of caveats. But otherwise, everything else, all active tissues are gonna have capillary beds. Why? Because remember, capillaries are the only place that anything enters and leaves the bloodstream. That's it. So if you're an active cell and you need oxygen and glucose or fatty acids or whatever to be delivered to you, then you need capillaries nearby. Arteries and veins will not do you any good. Arteries and veins are not going to deliver oxygen or carry away CO2. Nothing enters or leaves the bloodstream unless it's via a capillary. Remember when we talk about blood flow, that we have the blood, and then we've got our cells over here. What's the fluid that surrounds the cells called? Interstitial fluid. So when we start to talk about the diffusion of materials or the movement of things in and out of the bloodstream, we say that they go from the blood to the interstitial fluid, from the interstitial fluid to the cell. Okay, or the opposite way, from the cell to the interstitial fluid, like a waste, like CO2, from the interstitial fluid to the blood. That is, it seems so like, yeah, duh, but it's gonna be really important. When we start to talk about differences in pressures and osmolarity and how this stuff actually moves, it's going to be important that you realize we're looking at the interstitial fluid, not the intracellular fluid inside the cell. So again, capillaries are very, very thin. There's no tunica externa, no tunica media. Essentially, we're just looking at the tunica intima, right? This thin endothelium with an itty-bitty little basement membrane. What's the endothelium made up of? Endothelial cells. But what are the endothelial cells? Simple squamous cells. One layer of thin cells. This is an itty-bitty thin membrane. So the walls of capillaries are like nothing. Okay, like very, very, very thin, paper thin almost. Very, very thin. When we look at capillaries, we've got two general types of capillaries, and then we can even get more specific within them. But the two general types of capillaries that we'll talk about are continuous capillaries and fenestrated capillaries. So continuous capillaries, which are the most common capillaries that we have throughout most of our tissues and organs, they're called continuous because they have a continuous endothelium. Right, you're a complete endothelial lining without any big gaps or pores or holes in it. That endothelium it's nice. It's a pretty solid lining. Okay? This is important because that helps to kind of restrict what can enter and leave the bloodstream. It makes it harder for things to enter and leave if you don't have big holes and gaps present. Again, we see these in almost all active tissues. I mentioned before, not in the epithelia, right? So like your epidermis is avascular. And we don't have it in the center of cartilage, but it's in the membrane outside of cartilage, the perichondrium. So continuous capillaries. Again, these are the general common capillaries that we have in most places. So they allow the diffusion of water, of small solutes, so that would be stuff like ions and glucose, and then lipid-soluble materials. So remember, the respiratory gases are lipid-soluble. So oxygen and CO2. So these continuous capillaries that are located throughout almost every tissue in the body allow for all of these things to really easily cross in and out of the bloodstream. Okay, we've got channels that allow for the water and the small solutes to cross in and out, and there are small little holes between the cells that we'll find, really tiny. And then these lipid-soluble materials like oxygen and CO2, remember they can go straight through the lipid bilayer of the cells, right? They can go straight through the plasma membrane, and you have one thin cell. So it's really easy for those to just freely cross in and out of the bloodstream. However, because there are not big pores or gaps or holes present in the lining of continuous capillaries, they do not allow blood cells to cross in and out. Okay, so red blood cells are stuck. They're not going out into the interstitial fluid. They do not allow big plasma proteins to go out. Remember we said plasma proteins are stuck in the plasma. And they do not allow big, huge macromolecules, like some big nutrients that we would find in the GI tract, like some hormones, some really, really large molecules, large proteins are not able to cross these continuous capillaries. They're just too big. We have highly specialized continuous capillaries in a couple really protected areas in the body. We have them in the brain and we have them in the thymus. When we look at these specialized continuous capillaries, we see that the endothelial cells have tight junctions between them. Remember I just said that continuous capillaries don't have big holes or pores, but they might have itty bitty spaces between the cells? These specialized continuous capillaries don't. In between their cells, they have like desmosomes that connect them together. Okay, so they're really, really tightly connected together, like stapled and cemented between the cells, so that it's really, really hard to cross these capillary walls. Again, this is important because in the brain and in the thymus, we want to really protect these areas. We don't want things like medications, right, drugs or toxins or bacteria to easily enter these areas. So we make it really, really difficult to get in and out of the bloodstream. In the brain, we looked at this when we did neuro. This was what the blood-brain barrier was. Remember when we looked at the blood vessels in the brain, we said we have this blood-brain barrier. The tight junctions between the cells make it really hard to get into the brain or to kind of infiltrate that area. The reason we have it in the thymus, so the blood-thymus barrier, is there because T cells mature in the thymus and they have to be kept away from all antigens. When the T cells are maturing, they cannot see any antigens that are present. They don't want to see your antigens on like your red blood cells and stuff. They don't want to see any foreign antigens that enter your body. They have to be kept in this perfect little segregated environment while they mature. So it's important that we have these tight junctions in the capillaries there so that no antigens are able to actually enter into that thymus while the T cells are maturing. Okay. And this is why, like when you talk about medications, you'll say, oh, does this penetrate the blood-brain barrier? Does this have CNS penetration? Are there CNS effects of the drug? Is it entering the central nervous system or not? Is it crossing the blood-brain barrier or not? If you want something to cross the blood-brain barrier, you're going to change the drug to make it lipophilic, to make it like lipids. Because remember, lipids don't need gaps. Lipids can go straight through the cells. So it doesn't matter if you have tight junctions there. I can just cross the plasma membrane if I'm a lipid. So lipophilic drugs have better CNS penetration because of that. Fenestrated capillaries. Okay, so those were continuous capillaries, right? Most continuous capillaries let small stuff in and out, right? Glucose, ions, water, respiratory gases. The specialized continuous capillaries have very restricted permeability. Fenestrated capillaries have fenestrations. Okay, they have pores or small little passageways present in their endothelial lining. So because they have these pores present, they allow things to enter and leave the bloodstream more easily. And they also allow some larger things to enter and leave the bloodstream. So when we look at the normal stuff like water, ions, glucose, some little nutrients, those things really, really rapidly enter and leave the bloodstream. Also, we see that some larger solutes like amino acids, some larger kind of like disaccharides, larger sugar molecules, some hormones can enter and leave through these pores. Okay, so larger things, amino acids, hormones, nutrients like disaccharides. I didn't spell that wrong, don't hate me. Can enter and leave through these larger pores, these gaps. So we see fenestrated capillaries in multiple different places, and I'll kind of tell you why for each one. We see them present in some endocrine organs. What do endocrine glands make? The pituitary gland, the thyroid gland, what are they making in general? Hormones, right? They make hormones. Hormones are chemical messengers. Where do they travel through? The bloodstream. So these glands make these hormones. They're kind of big. Most of them are proteins, and they have to put them in the blood somehow. If you have a blood vessel that doesn't have any pores in it, how are you going to pump all these things into the bloodstream? You can't, right? You need a door. You need some way to get them in really rapidly. So that's why we have fenestrated capillaries there. Okay, so fenestrated capillaries allow these endocrine organs that make hormones, allow the hormones to enter the bloodstream through the pores. And the kidneys. So the kidneys are where we filter the blood, right? And we get rid of extra ions and waste products and all of that. We filter that stuff out of the blood and we get rid of it in the urine. When we filter the bloodstream, if we had continuous capillaries there, we couldn't filter out the wastes. They're too big. So we need fenestrated capillaries so that we have these pores so that the wastes can actually fit out of the bloodstream. Okay, so the pores have to be present so that the larger molecules like wastes can get pushed out of the blood and into the urine, right, into the waste. What do we do in the intestinal tract? What are we doing in the small intestine with nutrients? Absorbing them. After we break them down, we absorb them, right? We need some way, we bring them into the body, and then we gotta get them into our bloodstream. These nutrients, some of them are kind of big. Okay, we have some like amino acids. We have some fats that get packaged together in these little vesicles called chylomicrons. We have a lot of stuff that we need to really rapidly absorb into the bloodstream. These fenestrated capillaries make it easier to do that. The pores allow us to get water and ions and glucose really quick, and the pores are big enough to allow us to get some of those other larger nutrients into the bloodstream. Okay, so that's why we have fenestrated capillaries in these very, very distinct areas. Now, we also have very specialized fenestrated capillaries that are called sinusoids or sinusoidal capillaries. These don't just have pores present, they got big gaps between endothelial cells. Okay, so this is not a great barrier. There are really nice sizable gaps between the endothelial cells. And then the basement membrane is just not a barrier. Okay, it's either thin or absent. So it's really, really easy for things to cross in and out of these blood vessels, these sinusoids. We see them in the liver, spleen, and bone marrow, which those three things should sound kind of familiar from some other stuff that we've studied, the liver, spleen, and bone marrow. And then we see them in some of our endocrine organs. Again, we said that it allows free exchange, so really easy, rapidly the exchange of water, of ions, of pretty much any of the little stuff can just freely go back and forth. Look at this, it also allows the exchange of large plasma proteins. Okay, that's weird. We don't allow that to happen anywhere else. Okay, these are really big. So plasma proteins can enter and leave the bloodstream in these sinusoids. This is one of the reasons why it's important that sinusoids are in the liver. Where do we make plasma proteins? Huh? In the liver. So if we make these plasma proteins and we have to somehow put them into the plasma, we need doors to do that, right? We need to get them into the bloodstream somehow. So we have to have these sinusoids in the liver so that after we make the plasma proteins, we can put them into the blood. If we had continuous capillaries in there, we couldn't actually squeeze them in. We'd never actually get the plasma proteins into the plasma, okay? Also, what did we talk about when we talked about red blood cell recycling? Where did we say that these macrophages engulf red blood cells that are worn out? What do you know? The liver, spleen, and bone marrow. Okay, so macrophages are going to monitor the blood, and they need to engulf, okay, any old or worn out red blood cell in the liver, spleen, and bone marrow. Sinusoids allow that to happen. Okay, the sinusoids give the macrophages access to the red blood cells. Without those large gaps, they couldn't actually access those large red blood cells. The bone marrow, it's also important because remember, that's where we make blood cells. Okay, so we make the blood cells, we got to put them in the blood. So we need large enough gaps to actually do that. Okay, and we have that in the sinusoids. Okay, so bone marrow also is to, that's where we make blood cells. Okay, and again, endocrine organs, we talked about that already. They make hormones, right? And hormones are relatively large molecules. Some of them are very big proteins. So we need these large gaps in order to get them into the bloodstream where they need to travel. Again, we're talking about capillaries, right? Capillaries are the smallest vessels. They're also the most numerous vessels. They have really, really thin walls. Capillaries, how many layers do the walls have? One, right? What layer is that? The tunica intima, right? The tunica intima or tunica interna. I think your lab manual might call it interna. That's the innermost layer. And essentially it's just an endothelium with a little basement membrane. Right? The endothelium is made up of what kind of cells? Simple squamous cells. They're thin, right? Endothelial cells. So it makes for a very, very thin little membrane. That's why stuff can cross in and out of the bloodstream easily. Especially stuff like water, ions, and lipid-soluble things like the respiratory gases. Here you see a continuous capillary, which is the most common. Again, when you look at this, you don't see any big pores present. I said that there were tiny little spaces between the cells, and of course we always can have things cross through in vesicles, we can have things cross through in channels, and stuff like oxygen and CO2 can go straight through the cells because they're lipid soluble. But these are pretty restrictive. They're in most of our tissues. Fenestrated capillaries have fenestrations, right? They have pores that are present. And like when you look here, that's what all of these tiny little dots are showing you. Okay, then notice they're not present here. These fenestrations or pores allow us to really rapidly have things enter and leave the bloodstream. So that's important in areas like the kidneys. That's important in areas like the intestines where we have to filter out or absorb a lot of stuff really quickly. They also allow some larger molecules through. Okay, like some hormones, some amino acids, that sort of thing. Sinusoids have big old gaps between the cells. Okay, so again, they're still fenestrated. Notice you still see the fenestrations, but they're specialized. They also have these big gaps present. And those gaps are big enough to allow things like plasma proteins through. They're big enough to allow things like blood cells through. So that's where we have them in the liver, where we make plasma proteins. Where we have them in the liver, spleen, and bone marrow, where we monitor for old red blood cells. The bone marrow, where we make blood cells. We need some way to get those things in. So we do it through these big gaps in the sinusoids. Let's do like one more slide. We're ahead of where I wanted to be, so I'm happy. When we talk about capillaries do not exist as one single vessel. Okay, it's not like you have an artery, a capillary, or an arterial, a capillary, and then a venule. It's not like that. You don't have this one single vessel. Capillaries exist as capillary beds or a capillary plexus. So throughout this tissue here, you'll have all of these like intertwined vessels. So lots of different vessels that all kind of like interconnect with each other, and they go back and forth in all different directions. And so that you've got all these interconnected vessels. So it's not one single vessel that exists as a capillary bed. And again, that connects the arterial end to the venule end, right? The artery side to the vein side. We see that when we look at a capillary bed, we have numerous precapillary sphincters. A sphincter is just a circular band of muscle, right? Like the anus, the anal sphincter. Everybody understands it's a circular band of muscle. It can relax or dilate, and it can constrict or get smaller, right? So precapillary sphincters are circular bands of muscle that guard the entrance to a capillary. And again, they have the ability to contract and relax. And that allows us to control what part of the capillary bed the blood is going to flow through. This is really important because we need to kind of like constantly alternate the blood through different parts of the capillary bed depending on what each area of the tissue needs. So you could have a little precapillary sphincter right here, right? Another precapillary sphincter right here. And they can change the flow of blood through this capillary bed. So say this area over here was really low in oxygen, right? It needs more oxygen. So this precapillary sphincter could get bigger. More blood would now flow this way. Okay? If this area had plenty of oxygen, this precapillary sphincter could constrict because there's no use allowing blood to flow this way when we don't need it, right? It's better to divert it to another part of the tissue where we need it. So these precapillary sphincters are constantly contracting and relaxing in order to alternate blood flow through different parts of the capillary bed. We call that vasomotion. So collaterals are multiple arteries or arterioles that go to one capillary bed. Okay, like collateral is like outside, right? Like you put collateral, you put up collateral, you put something else on the table. Collateral damage is like damage that's outside of the intended damage, right? It's like something extra. So if we have like one capillary bed right here, I can have multiple vessels that lead to it. Okay, those are collaterals. This is important because you can maintain blood flow even if you block one of them. So say there's damage or a clot or something that blocks this vessel, you still have blood flow coming in from this side. So you're still going to bring blood into this capillary bed. And so it allows for two different routes of blood flow. So if you block one, that tissue is not going to die. You still have an alternate route for the blood to come in. An arterial anastomosis is a like where the connection comes between the arteries. So where like the two collateral arteries come in to form an arterial, that's the anastomosis. That's the connection between the two as they fuse together. An arteriovenous anastomosis is a connection that goes directly from the artery to the vein, and it skips the capillary bed altogether. So we can have another vessel that comes down like this, goes past, and then comes up into the venule. And it completely skips that capillary bed. And sometimes we need that to happen. We might have like a hemorrhage or an emergency where we're not getting enough blood to the brain. So at that point, quite frankly, we don't care about blood anywhere else. We'll dilate these the arteriovenous anastomosis, make it really big, so it's easier for the blood to come here, and the blood will skip all these other tissues so that we have more of it to go up to the brain. Because we just kind of skips that capillary bed. I'm sorry, I'm giving you guys an extra couple minutes, but we're so close to being done. That makes me really happy because that puts us ahead. So we just need a few things, guys. This is backwards of how I always draw it. So I always do like arteries to veins, so just notice it's backwards. But you see the arteries coming in, right? These are collateral arteries because two of them are coming in towards the same capillary bed. The anastomosis is where they connect right here to form the arterial. right? So the oxygenated blood's coming into the capillary bed. We're delivering it to all these cells that are in the tissue. We make our way over here to the end of the capillary bed, and that brings the blood into the venule, the smallest little vein. The venules are going to merge to make bigger veins as we carry that deoxygenated blood back towards the heart. You can see precapillary sphincters located all over the place. These again are like circular bands of muscle that will control where the blood, actually this will go this way, that will control where the blood is actually flowing. Okay, they circle it. Okay, they can get bigger to allow blood to flow that way. They can get tighter if we don't need any blood to flow in that direction. This is the arteriovenous anastomosis. Remember, this vessel can completely skip this capillary bed. So if I don't need the blood here and I drastically need it somewhere else, I can make this vessel get really large so there's not very much resistance. So it's easier for the blood to flow this way and just completely skip the capillary bed and move on somewhere else. Last thing, I mentioned vasomotion. I mentioned that the blood constantly changes route through the capillary bed, right? Different areas of the tissue, different cells might need more blood at different times. So we tell those precapillary sphincters to contract or relax to constantly alter that blood flow and bring it to the side of the tissue that needs it most, right? To deliver oxygen or to pick up CO2 and carry away waste as needed. So the blood's constantly kind of changing routes through that capillary bed. That's vasomotion. Oh, sorry, I really want to get through it. Do you guys have any questions?